Okay, so now let's start the meeting. Good morning and good evening, dear experts. And I'm Andrea Chunshi from Clinical Surgical Oncology Editorial Office and GMed. Thank you to all the distinguished experts today. And we are holding the fifth ESSO-CSO academic webinar in 2026, and today we will focus on clinical research training, building surgical research within the ideal framework from preclinical studies to randomized trials. And before the academic session, I would like to take a few minutes to introduce CSO. CSO is hosted by Jiahui International Hospital, and it is seeking innovative research and advancements in surgical oncology. The dedicated efforts of editorial board members have created a very high quality academic exchange platform for us. And our article downloads in 2025 exceeded more than 12,000 times. Thank you very much. And let me introduce our today's moderators. Dr. Nada Sanchek. She's a general surgeon at Institute for Oncology and Radiology of Serbia. She is also assistant professor at Medical School, University of Belgrade. And the other moderator is Dr. Qiuchen Zhao. He is a postdoctoral fellow, Yale School of Medicine, and honorary trust scholar, School of Clinical Medicine, University of Cambridge. And next, let's welcome Dr. Nada to be our moderator. Thank you very much.
ESSO is a diverse and inclusive community fostering connections among surgical oncology experts worldwide. ESSO community counts 4,500 plus members, global membership with 25 national affiliated societies. Our mission is to advance the science and practice of cancer surgery through education, research and innovation and multidisciplinary care in Europe and beyond. Through our courses and webinars, we train surgeons and healthcare professionals on the latest advancements in their field, new surgical techniques and surgical research. Our specialized European schools offer structured training in the management of peritoneal neoplasms, soft tissue sarcoma surgery, and breast cancer surgery. With a passion for innovation, our research academy aims to promote surgical oncology research and collaboration among healthcare professionals, researchers, and patients. To support the next generation of surgical oncologists, a growing network of young ESSO was established to connect junior surgeons and help them play an active role in the future of surgical oncology. Through our educational grants, we provide young ESSO members with opportunities for learning and research development. With this vibrant community of dedicated professionals, we organize the biggest annual surgical oncology congress in Europe to stay at the forefront of innovation. Our commitment to advocacy ensures that the voice of surgical oncologists is heard, influencing decision makers and shaping healthcare policies. At ESSO, we unite surgical oncology experts across the world, all with one common goal: enhance the quality of cancer care, cure cancer together. Follow us at essoweb.org.
Thank you for your kind introduction and welcome from my part as well to today's webinar. It will be my pleasure to co-moderate with a colleague from Yale University, Dr. Zhou. To start today's journey, we have prepared a few introductory slides on ESSO to get you acquainted with what our community is dedicated to and what it has to offer to our members and international friends and supporters. As you see, our unique mission that it has been recognized by over 4,500 members all over the world is to exchange knowledge, to inspire education, research, and especially encourage high quality research. Also to campaign for the harmonization of education across Europe, to standardization of surgical oncology training, to stimulate support and innovation in surgical oncology, to promote the role of surgical oncology in multidisciplinary care, to share our experiences and knowledge. There are many benefits of becoming member of our society. You can benefit from reduced rates to our congresses and other educational activities, such as this webinar and other hands-on courses that we are organizing in high quality. There is also a very important advantage in publication because ESSO subscription is significantly lower for people who are members of our society. And there is also a 30% discount for the publication in ESSO. Another important benefit is EBSQ examination in surgical oncology, which is a high-level recognition of knowledge, skills, and professionalism at European level. It is organized by Division of Surgical Oncology of the Section of Surgery of the UEMS with the support of ESSO. And every year, along with the Congress, we have these examinations. So after this qualification, you become a fellow of the European Board of Surgery, which is highly recognized in many countries in Europe and also world. Next slide, please. As I said, there is a benefit for publishing in ESSO, which is the official journal of our society and BESO, and also very important discount fee for the publications. Next slide, please. I would kindly invite you to join us in our efforts to make surgical oncology belong to everyone, to be without borders. This year's topic is innovations and challenges worldwide. So contribution from our Chinese colleagues will be significant. Our 45th Congress is held in Madrid from 4 to 6 November 2026. And you can check the registration fees and the discounts that I've been talking about for all the membership of ESSO. Next slide, please. Now we continue to the webinar itself. We have designed it together with the CSO colleagues, and it will be focused, as they said, on surgical research. Why? Surgical oncology is increasingly driven by evidence-based practice, yet generating high-quality evidence in surgery remains uniquely challenging. Unlike drug development, surgical innovation often involves complex interventions, learning curves, technical variability, and difficulties in conducting randomized trials. The ideal framework was developed to provide a structured pathway for evaluating surgical innovations from preclinical and early developmental stages through assessment and long-term implementation. And today we will explore how surgical research can be strengthened across this continuum. And the next 19 minutes, we will discuss how surgeons can contribute to the generation of robust evidence that ultimately improves patients' outcomes. We hope this session will stimulate further collaboration between clinicians, researchers, statisticians, and trainees, and inspire the next generation of surgical oncology research. Our speakers today, as you see in the slide, will be Professor Chu from Shandong Cancer Hospital and Institute, and also professor at the First Medical University in Jinan, China. Next speaker will be Dries Reinders, statistician from Ghent University in Belgium, and Professor Shiraz Marker from Oxford University Hospitals NHS Trust, Oxford, UK. You are free to share your comments and ask questions in the Q&A section and also in chat. So we can start with our first speaker, Professor Chu, who will talk about breast cancer preclinical studies on the model of lymph node metastatic immune remodeling and immunometabolic therapy. Professor Chu, the floor is yours.
Hello, Dr. Zhou. Hello, Dr.
Yeah, our next speaker is Mr. Dries Reinders, a statistician from Ghent. He will talk about causal interference for surgical oncologists. So the floor is yours, Mr. Reinders.
Thank you. Good afternoon. Voila. I'm assuming everybody can see the slides now. So I'm here to talk about causal inference, an introduction to causal inference. I'm a statistical consultant involved in clinical trials in PPAC, also involved in observational studies, but not in the field of surgical oncology. And let's dive into it immediately. So in general, we can divide the goals of medical research in roughly descriptive research, predictive research, and causal research. Descriptively, we can look at tumor burden over different ages, patient characteristics at diagnosis, and that's information that is relevant. But it doesn't really describe the causal effect or anything. The next step, we can try to make predictive models so that if patients enter your office, you can say, well, given your characteristics, based on what we know, you have this probability to survive the next 2 years. This is predictive and doesn't mean that specific inputs into the prediction model that you have identified causal effects of age, of tumor burden, and so on. It means you can use them to predict without guarantee that they are actually the cause. And this is a large part of medical research, causal questions. What is the effect of, for instance, a surgical treatment versus non-surgical standard of care on overall survival? Of course, you also have causal questions on etiology. If we apply this treatment, what does it change in the body so that it impacts survival or progression or things like that? That's not the type of causal effects I will talk about today. Today, it's mainly about comparing treatments, comparing the impact, assessing the impact of interventions. And then the first thing when targeting a causal effect is that it's important to be explicit on that. Explicitly state that you are targeting a causal effect. Not just we look into factors associated with, to then in the end, in the discussion, use all kinds of causal language. No, if you want to know a causal effect, clearly state you're targeting a causal effect, and of course adapt the analysis to these goals. And for such analysis, you need more than statistics. And you can find in data, you can find associations. To interpret these associations as causal, you need additional assumptions that are often untestable, or at least, or maybe only partly testable. And hence, to know whether the assumptions we make are reasonable, we need subject matter expertise, which will not come from the statistician, but from you. And then the goal is something like, look, we here have data on people treated with treatment A. Let's call it cohort 1. We have data in cohort 2 treated with B. And now, can we learn from cohort 2 what would have happened with cohort 1 if they got treatment B? That's a typo there. Can we learn what would have happened with cohort 1 that actually received A if they would have received treatment B? If so, then we can contrast it, we can compare it, and that comparison reflects a causal effect rather than just an association. And to understand the assumptions that you require and how you can think about this, randomized trials are a perfect beginning. Because there it's very, very clear that you are targeting a causal effect. And the reason that randomized trials are so great is that they, by their design, they make many of the required assumptions to interpret an association found as a causal effect, that they make these assumptions much more likely. through the randomization, through through clearly defining the treatment and the surrounding care, and when and how outcomes are measured, and so on. If you look at the drawing at the bottom, we have those randomized to treatment A in blue, those randomized to treatment B in red. And then what we can do in practice is compare the outcome in the blue with the outcome in the red. Why does this work? Because thanks to the randomization and all these other measures, it's reasonable to have the outcomes in the red at the left bottom, to see these as a stand-in for the outcomes of the blue in a parallel universe at the bottom right, if they would have gotten treatment B. So the comparison we want to make is on the diagonal, blue top left, blue bottom right. But since they are since they occur in parallel universes, we can't really compare these. We don't have the data from the parallel universe. Similarly for the red, of course. And by the randomization and all the others, the right at the left, the observed results in the right, in the red, are a good stand-in for the ideal results in the blue at the right. So this is in a randomized setting. What if we go to a observational setting? If we have observational data, can we then still learn what would have happened with cohort 1 if treated differently from the data we have available from cohort 2? Lots of potential issues arise: confounding, selection bias, misaligning the time zero, measurement error, all things that can lead to associations showing up while in practice there are none. Or for an observed association to be different from the real underlying causal association. We can't discuss everything in depth, of course. We only have 20 minutes, but well, let's look into confounding just for a few minutes. If you look at a at a graph at the bottom, that's called a DAG, directed acyclic graph, and in that graph, I sketch a very simple scenario in which disease stage influences both treatment selection and the outcome, and the same for SES. You see nothing beyond this, so within this scenario, we assume that's all the causal impacts that there are. There are no other unmeasured factors that also influence the treatment, that also influence the outcome, or anything else that is drawn here. Now, what this means if disease stage impacts treatment and outcome, it means that if we compare a group treated with surgery and another group treated with chemo only, if we observe differences in outcomes, that maybe the observed differences are due to differences in disease stage and or differences in SES, rather than due to an actual impact of surgery versus chemo. I've tried to visualize this on the next slide. So we have an outcome on some scale where higher is worse. And then I made a graph within each disease group. I ignored the SES for now, just to make it slightly less complex. So within disease stage 1, you see that treatment A tends to have higher, worse scores compared to those treated with B. Similar for the greens, similar for status 3, and similar for status 4. But what you also see is that as the disease status increases, so tend to do the outcomes. And in disease status 1, most are treated with treatment A. In disease status 4, most are treated with treatment B. So while within each disease status, treatment B shows it's doing better than treatment A, overall, if we simply compare the crude outcomes, treatment A versus treatment B, the outcomes tend to be worse for treatment B because they have more of these disease status 4 patients. How can we deal with this? There are multiple options. Two of them, I addressed two of them here. First, we can just compare within strata. So if we assume the only confounder is this disease status, we can look within DS1, within DS2, within DS3, within DS4, and then somehow pool these results. And then you see in each of the strata, it points to the same direction. Treatment B does better than treatment A. And this is essentially what you do with a multivariable regression model. If you have a regression model, outcome, treatment, and disease status, then you're estimating effects conditional on being in the same. You're estimating differences conditional on having the same disease status. An alternative that occurs often is matching. Link a patient with specific characteristics treated with A with a patient with as similar as possible characteristics treated with B. Make such pairs, and then on the whole, you are comparing likes with likes, and you, as such, removing the confounding. Of course, if we know, as in this setting where I made up the data, that disease status is the only confounder, then that's not too difficult. In practice, we don't really know all the confounders, and we haven't all measured them, so it's much more complicated. Another technique that I will spend a few slides on quickly explaining how it goes is propensity score weighing, where we create a pseudo population where confounding no longer exists. So let's say this is what we have. We have a group treated on a control treatment, a group of patients treatment with the experimental treatment, and one confounder, one characteristic that is important, namely blue versus red. And then you see that the relative proportion of blue in a control group is higher than in the treatment group. Hence, if we compare the outcomes, the distribution of the outcomes in the control and in the treatment, and we compare them, for instance, by comparing averages, if we see a difference, maybe it's because of the disproportionate presence of blue people instead of because the actual impact of the treatment. As we just said, we could compare the blue with the blue and the red with the red and then somehow pool. Here, we're gonna take a different road and weigh them. So what you can see is we have 8 blue in total. Only 2 of them are in the treatment group. Probability of treatment, 2 out of 8. We have 6 red in total. Only 2 are in the treatment group. 2 out of 6 is the probability of being treated with the experimental here. And now we assign weights. So instead of counting 1, 1, 1, 1, 1, 1, 1, 1, 1, each person here counts for, each blue person here counts for 8/6, while each blue person here counts for 8 seconds, for 4. And similarly for the red, each red person counts for 1.5, 6/4. Each red person here counts for 6/2, 3. And then you have 6 times 8/6, and so on. And then you see in this weighted pseudopopulation, you have an equal number of weighted blue and weighted red. And as such, the confounding is dealt with. You have groups that are balanced on blue versus red. And if we then in that weighted pseudopopulation see a difference in average outcome, it will no longer be because of different proportions of blue versus red. So if the color is then, in this case, the color is the only confounder, then in this weighted pseudo population, we can estimate an actual causal effect. Many other biases exist. I'm gonna list just a few. Let's say that in our observational data set, we only know about the treatment decision not at the time that it's actually made, but at the time of actual treatment initiation. In that case, maybe we have lost a few patients that died, where the decision was taken, for instance, to undergo surgery, but they die before the surgery could actually take place. And hence we can't identify them, we lose them. They're not in our analysis data set. Another impact is that maybe for surgery, it may take some time to actually take place, while the competing treatment can be initiated almost immediately. And then, of course, there's a structural difference in timing from actual treatment decision where time 0 is taken, which will bias comparisons. You can also think, if you're thinking about selection bias, it can also end up as missing data. If those where you don't have the outcomes for tend to be different on certain characteristics that impact the outcome, then in the end, the data you are working with will provide a biased comparison unless you are able to adjust for it. And a framework I want to very quickly present here in the last 2 minutes is a target trial emulation framework. If you could, what randomized trial would you conduct? Write out a protocol for that randomized trial, and then, given the observational data you have, how can you mimic this as good as possible? I have an example here from a surgical treatment, but not in oncology. But you can see here eligibility criteria, treatment strategies, assignment, time 0, and so on. For instance, time 0 in a randomized trial is, of course, the time of randomization. In the emulated target trial, if you have access to that information, it can be the time of the multidisciplinary meeting where the patient was assessed, which is probably indeed the time where the treatment decision was taken. Often this kind of information is not available, and you'll have a slightly worse or significantly worse proxy for this. Similarly, for eligibility criteria, they are very not easily assessed in a randomized trial, but it's much more difficult to fine-grainedly assess eligibility criteria in an from observational data. But this kind of approach to guide your observational analysis with a causal question can help to identify biases like those listed here. For instance, in a randomized trial, it would never you would never allow to have vastly different assessment times in one treatment group than in the other. Well, maybe in your observational data it is. And The target trial emulation framework helps you think this through. And then, of course, a next step is actually address this and hopefully solve this slightly. That's all for now. I have a few resources that can be of interest, a few really good books. What If from Hernan and Robbins, The Effect, Nick Huntington Klein, The Book of Why, and something I really like, the YouTube course of Rachel McElrath, Statistical Rethinking, which is heavily Bayesian, but also has an important causal component to it. And then some R packages that can be useful, but I think it's time to stop for me.
Dr. Zhao, so please introduce the next speaker. Thank you.
Hello, can you hear me?
Yes, we
Okay,
can.
yes. Let's welcome Professor Shiraz Malka, who's the professor of upper gastrointestinal surgery and also the director of the Surgical Interventional Trials Unit from University of Oxford. So today he's going to present the topic on how do we improve the surgical trials in 2026. Let's welcome him.
Great. Many thanks for the opportunity and the privilege to present here today. So I'm going to talk to you really about where we are and potentially where we may be going with surgical trials in the future. So these are my disclosures. So I'm director of the Surgical Interventional Trials Unit in Oxford, and we have on our portfolio around 27 randomized controlled trials. These are really across a broad portfolio of surgical specialties, including upper GI, lower GI, breast, thoracic, transplant, urology, and neurosurgery. We run an annual budget of around 27.5 million. Which is large for most surgical trials units across Europe and across the UK, definitely. Most of our funding comes from our government in terms of national funding. So I put lots of pictures of my children in my slides, but essentially I think there are currently three main challenges with surgical trials. I think they often lack external validity because we don't have a really clear approach as to what happens after the trial, basically. I'll talk a little bit about this. Secondly is, when do we actually do the trial? When do you think it's the right time to do the randomized control trial in terms of the evolution of the technique? And I'll talk a little bit about that as well. And then the most important thing is as the previous speaker alluded to, is the randomized design actually applicable to all surgical innovations, i.e. surgery is not a drug, and we have to start to get out of this mindset of treating it like a drug. So when we think about whether or not surgical trials have external validity, I often think of it a bit like taking a picture with your children, whereby you take a really nice picture, it's in a nice environment, everyone looks happy. Just like when you do your trial, you do it in a really nice group of centers. They're probably the best centers in your country. And the question is that really representative of real world clinical practice? And the answer is it can be variable. And that's honestly what we see when we start to think about how these things work. So this is a good example of this. This was a paper I published a few years ago now in JCO. But essentially, we looked at the outcomes of minimally invasive esophagectomy. So the figure on the left shows the outcomes within the TIME trial, which is a randomized controlled trial conducted in Netherlands of minimally invasive versus open esophagectomy. It's performed in 6 centers, and they showed a massive reduction in pulmonary complications associated with a minimally invasive approach to esophagectomy. This led to a huge uptake of minimally invasive esophagectomy, not just in Netherlands, but internationally. So then what I asked them for was, I asked the Dutch for their data immediately after the trial, for the following 6 years after the trial, to see what the results were when the technique was applied nationally in the same country where the trial was conducted. And what you saw was that actually a minimally invasive approach was associated with an increase in pulmonary complications. So therefore, that trial has absolutely no external validity in the country where the trial was performed, which is a major issue when you think to really what is the point of doing these randomized controlled trials in many ways. The other thing that we can't get away from is that I work in the field of upper GI surgery, and mainly specializing in esophageal and gastric cancer. And there has been no interventional trial whereby the intervention is a surgical intervention in my surgical specialty, which has led to an improvement in overall survival for these patients. And we are really in the business of trying to change overall survival, and yet we've not been able to do that in any trial whereby the intervention has been a surgical intervention. And I believe that what this is really because the observed variability of what we see in surgical performance actually varies so much between centers and surgeons that it ends up overpowering the actual intervention within the trial. And this is a real major issue when we start to think about how we can actually apply evidence within this context of patients and recommendations moving forward. And the thing that we have to really start to understand is that surgery is not a medication, and therefore there will be variability in performance. And really this comes down to many factors, but one of these is proficiency gain curve or learning curve. So I published this a few years ago, but essentially we looked at our national data in the UK for modeling learning curve with the introduction of minimally invasive esophagectomy and gastrectomy. And what we showed was that when you introduced minimally invasive esophagectomy, if you were in the early part of a surgeon's learning curve, your mortality rate was double that was seen in the latter part of the learning curve. And that is just mortality. And I believe that is unacceptable in 2026, in my opinion. The second aspect of all of this is when to do your trial. Do you do it in the early phase when it's exciting and really it's you're probably going to get your Lancet or New England paper, but in reality the technique is probably under development, so we're sort of in an ideal phase 2A in reality. But you try to standardize the technique, and often what you find is in actual fact the surgeons are probably in their learning curve. Or do you wait too long, in which case the trial will become obsolete because surgeons will have adopted the technique anyway? And we're starting to see this a little bit with robotics, whereby surgeons are taking up robotic surgery with very little evidence in support of it, but yet they think it's the right thing to do. So if we were to do a trial on robotic versus anything else, it may be obsolete by the time the trial is complete. Classic example of really timing of trials, I think, is the logical trial. So again, a Dutch trial of laparoscopic versus open gastrectomy. What's important with this trial is that it showed no benefit to laparoscopic gastrectomy over an open approach in terms of length of hospital stay, which is the primary outcome, complications, or any oncological outcome. So this trial showed no benefit to a minimally invasive approach. Keep that in mind. Now, I asked the Dutch again for their data around the time of the trial. So the solid gray box represents the timeline of the trial, and the solid black line represents the uptake of a laparoscopic gastrectomy. What you see is there is an exponential rise in the rate of minimally invasive gastrectomy in Holland. Huge, basically, before the trial, during the trial, and after the trial. What you also see in the solid blue line is that in actual fact, the complication rate from laparoscopic gastrectomy dipped after the trial. So perhaps the trial was negative, but surgeons were actually in their learning curve during the phase of the trial, which is fascinating when you start to think about the timing of when to do a trial. I also wanted to really talk about variability in surgical performance. And I think what this really comes down to is that we often think of surgery and we capture surgery as an operation note. And I often think the analogy is like a piece of music. If you just take an operation note, it's just like having a piece of music. But we know the way that music is played sounds extremely different dependent upon the musician who's performing that piece of music. Likewise, a recipe for a cake is very different dependent on how you want to bake your cake. And that's exactly the same with surgery. So an operation note probably doesn't actually tell us how well the operation is being performed. So we published this again a few years ago, really looking at quality of surgery and how that was controlled within randomized controlled trials. This is across all oncological trials in upper GI cancer surgery, whether or not the intervention was surgical. So it includes chemotherapy trials as well. And what we showed was that if you had some aspect of surgical quality assurance, so i.e. if you credentialed your surgeons before they entered the trial to make sure they were good enough to enter your trial, your in-hospital mortality was reduced within that trial independent of any other factor. So therefore, it's a major component of not just surgical trials, but also oncological trials in terms of assuring that surgeons are of sufficient quality to enter the trial. And there are three phases to really surgical quality assurance within randomized controlled trials. Firstly, you standardize the technique, and that typically is done by bringing a group of expert surgeons together to do a Delphi consensus process, and you identify which steps are mandatory or absolutely should be done, which steps are optional, which steps should be prohibitive and should be not really allowed in actual fact. Then you credential your surgeons to make sure they're of sufficient experience to enter your trial, either by looking at their case volume or perhaps more sensitivity, as we're doing in our trials now, is whereby we ask surgeons to submit videos. We score those videos using the standardized technique, and then we allow them to enter the trial when they've reached a certain level. And then you also monitor the performance during the trial, and you constantly feed back to surgeons as they go through. So we developed these competency assessment tools in Oxford within randomized controlled trials. And I think they're really important because what you do is you identify the technical errors within the trial that can be linked to adverse patient outcomes. So you identify which steps of the operation you think are important. You look at the errors of those steps, and then you have the granular data at the end of your trial to say, okay, these steps are actually important. So therefore, when you start to think about external validity, you've developed a competency assessment tool now for your new technique, for example, robotic gastrectomy. And you say, okay, if you're good enough, you have to submit 2 videos to perform robotic gastrectomy independently, because this is the acceptable standard as defined in our randomized controlled trial. So you don't have this problem with external validity after your trial because you have a very well validated competency assessment tool. So we've started to do this within robotic gastrectomy, robotic D2 gastrectomy. This is led by one of my PhD students, Riyad Salem. And essentially, we did a Delphi consensus process looking at distal gastrectomy. We gained a consensus on 62 steps of the operation that should be performed. Likewise, in total gastrectomy, we gained consensus on 57 steps of the operation. What I'm interested in is that I think we always focus on the end product in surgery, whether or not the lymphadenectomy was done well enough or whether or not the anastomosis looked good enough at the end. But in actual fact, when you think about it as a surgeon, there are really important 3 other aspects that are really important. One is how do you expose the tissue? The second is whether or not you execute the task very well. And thirdly is any adverse events that occurred during that task. So what I'm trying to do is say, yes, we need to look at the end product for the context of the trial. Perhaps if we look at these other 3 domains, we create a platform whereby it's an educational platform as well as being a platform for surgical quality assurance within the trial, but it can be used for the implementation phase after the trial. So therefore, what we do within the Master D2 tool is that we also look at exposure, execution, and any adverse events. And we also look at the end product quality, as you would expect with most surgical quality assurance tools. And for every step of the operation, we provided photos to say what the end product quality should really look like in terms of lymph node dissection in this example. The second question is this is obviously very laborious, but the question is really, how do you meaningfully integrate surgical quality assurance into the life cycle of a randomized controlled trial? So what I believe is that this is a paper that we published really as a an opinion piece in the Annals of Surgery this year. And what we really thought about was really what you do in the pre-trial phase is you standardize the technique. So you go through that Delphi consensus process. Then you credential your surgeons by asking them to submit some videos. You make sure the videos are good enough and they're good enough to enter your trial. And then within any trial, there's always an internal pilot phase. And typically what we're trying to do in that trial, in that phase of the trial, is you're trying to say whether or not you can recruit patients to the trial. But I think you can collect that data and use it in a more meaningful fashion. So what I think you should be doing is during that internal pilot phase, is you should be scoring the videos. So rather than just saying, okay, what we've done is we powered our study on 100% of the surgeons performing a D2 gastrectomy to an acceptable level. In actual fact, what you'll know is that 80% of the time there's variability in performance. So within your internal pilot phase, you capture that variability in operative performance, and then you redo your sample size based upon compliance to the protocol. So that creates a more meaningful sample size, which is more reflective of what you expect to see when you apply this technique at a national level or an international level. And the elephant in the room is how does AI really help with that? So we've started to do some work really around AI and particularly using gallbladder surgery. So in the UK, we perform around 73,000 cholecystectomies in the UK. We audited for the last 3 months within Oxford. We collected all the interrupted videos of emergency cholecystectomy. And we showed that critical view was only obtained in 62% of cases. Now what we're doing is we're asking expert surgeons to look at those videos and say what they would have done differently. And what we do with language process modeling afterwards is that we create a tool whereby, as a trainee surgeon, if you are operating and you're having some difficulty, the AI coach will provide you with a couple of hints, which will say, I would do this, or I would potentially do this. And that may, in actual fact, raise that number of critical view that we're obtaining from 62% to a more acceptable level, I think, in the future. We're also doing some work with the Republic of Korea, and there's a joint collaboration between the UK and the Republic of Korea, really looking at whether or not AI can support surgical quality assurance. And one of the things that I think is really exciting within the context of clinical trials is that we're using AI in this platform across 3 phases. The first is, as you would expect within surgical quality assurance, 1, to recognize the phase of the operation, 2, to automate the scoring of the operation to make sure that it's acceptable and within the master D2 framework that I talked about. The second is whether or not AI can actually identify patients from the patient record, which would be eligible for inclusion in your trial, which would substantially reduce the burden upon the research nurses. And then finally, whether or not AI can collect clinical reporting outcomes from the trial. So it becomes an AI trial across the panacea, across basically both preoperative identification, intraoperative quality assurance, and postoperative collection of outcomes. And then finally, I'm the research methodology lead for the IDEAL framework. And one of the things that we said when we originally designed IDEAL was that surgical quality assurance is important. But we really gave no insight into how it actually could be integrated within the framework. So we've gone through a NIHR-funded Delphi consensus process really around how this should be staged. And what we've created is, and this is shortly due to be submitted for publication, but is really looking at when you should look at those 3 phases of surgical quality assurance. So standardization really goes up to stage 2B in terms of when you should be really looking at that. Credentialing of your surgeon, again, is from stage 2B into stage 3 for the assessment. Monitoring of performance goes through from stage 2B to stage 4. And in more granular data, we've also provided exactly what you should be trying to look for as a chief investigator in terms of task analysis at stage 0, trying to understand how you standardize the technique in a meaningful fashion. Also in stage 2B, how you actually disseminate this technique safely across centers. And then finally in stage 3 and stage 4, how you look at aspects of performance drift that may occur when you implement this technique at a national level. We also really look at potential challenges associated with this, and also really looking at potentially the utilization of AI in each of these steps as you go through the IDEAL framework. So I think to sort of bring this all together, I think there has to be a real clear line of sight from your randomized controlled trial to clinical implementation. Surgical quality assurance should be really used to define the quality of surgery and actually also standardize the intervention. The key questions in my mind are always around, if you control the surgery to such a level, does it actually limit the ability to perform the operation with a degree of variability which should be acceptable within surgery? And that's not what we're trying to do. What we're trying to do is that during the innovation phase of surgery, is that patients don't experience patient harm. And particularly within a randomized controlled trial, is that the validity of the intervention still stands. So therefore, I always think that when you design a randomized controlled trial, you must be really thinking about the clinical implementation that occurs after the trial. And I do believe that surgical quality assurance, with the development of competency assessment tools, actually will facilitate that implementation phase after the trial. So I'd like to thank you for your time, and I'll stop there, and we're very happy to take questions during the discussion.
Wonderful. Thank you for a very interesting talk. I think there is one question that can be answered by you. Is the video assessment also done as a regular quality check for surgeons or just during studies?
So at the moment, it's done largely speaking within studies, but within the UK, we had a national training program for laparoscopic colorectal surgery called LAPCO, whereby this was done in routine clinical practice. So yes, it's coming, and I think AI will help with this because obviously scoring all of these videos is quite laborious and it takes a long time. But with the utilization of artificial intelligence, even for surgical phase recognition, we take a 2-hour video and we're able to reduce it to 8 minutes of analysis. So this is really, really, I think, an exciting time for us.
Absolutely, I agree. Since there is no questions in the Q&A section, we encourage you to put down whatever comes across your mind, comment on the speakers' talks or some questions that remained unanswered at today's webinar. And just to start with the discussion, maybe I would ask all the speakers to tell me, is the ideal framework sufficiently adopted in surgical oncology currently in our clinical practice, or do we still mainly rely on traditional research paradigms, or what is your thought on this? Maybe start with Dr. Marker.
Yeah, so I think the ideal framework is it was never built to be a framework that stood still. So we are constantly evolving. We're just written a guidance on robotic surgery, which is about to come as well. And likewise, we've tried to integrate surgical quality assurance within that. So it will evolve as things go through. I do think it does provide us with an idea as we innovate in surgical oncology, how we should be doing that. And I think it really takes you through those stages and trying to put patient safety at the forefront of all of this, that really you shouldn't jump to a phase 3 trial until you've done your phase 1, your phase 2A, and your phase 2B. The question in my mind will always be, and this is a question for the panel as well, is whether or not the ideal framework is too laborious and whether or not it stalls innovation to a certain degree. And I don't have an answer for that. So very happy to discuss that.
Well, what do you think is the biggest barrier to producing high-level evidence in surgical oncology today?
So for me, I think at the moment, the biggest challenge in many ways is time. It takes a long time to conduct a phase 3 trial. If you look at a lot of the trials that I alluded to from the Dutch, I would describe them as phase 2B trials. They're not phase 3 trials at all. They're not, the sample size isn't big enough. So if you want to really provide robust evidence, you're talking about a phase 3 trial. I personally think, and I would be really interested in Professor Render's thoughts on this, is that we need to start to pivot towards platform trials and potentially embedding them within registries. So registry-based randomized controlled trials with an idea that these are more reflective of real-world practice and allows us to test multiple interventions within the same context or the same disease state within a similar platform.
Dr. Reinders, Mr. Reinders, actually, what is your thought on this?
Well, I think it's a very good suggestion at a kind of broader trial types, and the kind of trials that are run within registers, if the registries, of course, are of sufficient quality, because that will be a major issue for proper comparison of outcomes. You need very well and very well standardized outcome assessments. But if that is the case, it definitely can help to speed up things and indeed to perform trials in a population that is more representative of the eventual population that might receive the intervention.
The other aspect that I think is really important in surgical oncology, and alludes to the first speaker as well, which is that we have to use trials as an opportunity for translational research. There's no point. All of my trials have a translational component to them. We collect biosamples. We basically use them for our translation, because you're never going to get this opportunity again with really robustly collected clinical outcome data, as well as the opportunity to collect all these biosamples, which should be used for translational research. Absolutely.
My question for Mr. Reinders, statistically speaking, what methodological mistakes do surgical oncologists mostly commonly make when interpreting data? What is your view on this?
Well, let's say I don't pretend to know the average or even a reasonable sample of surgical oncologists. My work in surgical oncology is actually limited to a collaboration with one professor here at in Ghent. So I think, yeah, thinking of randomized trials in surgery in general, it's a lot of issues can be thought of as properly identifying in and exclusion criteria at a proper moment. I have seen more than one suggestions to evaluate a criterion at the time of the operation, post randomization. Things like that, of course, can really influence the results and the meaning of the results, especially if it occurs in one arm and not in the others. So of course that's part of the major complexities of running trials, comparing surgery with non-surgical interventions.
Yeah, I do think I often wonder. I really liked your thoughts on causal inference, because I think as surgeons we imply causation a lot from our data. It's not always there. And I think the other thing as well is that I talked a lot about surgeon variability of surgeons, but we also have variability of patients, whereby within our trials the patients are pretty tightly controlled, and then we try to apply this to the general population, which is really where we go from phase 3 to phase 4 within the ideal framework, and commonly it fails. Basically, it does, because the trial is not truly representative of the population that is having the surgery nationally, at least. It's not like a pharmaceutical trial in many ways, whereby there's variability also at the surgeon level, but very much at the patient level as well. So this is also a major Achilles heel, I think, at the moment of surgical trials.
Absolutely. In that context, how can the international collaborative networks improve recruitment and trial quality? Maybe Professor Marker can comment on that.
Yeah, so I think we have to be less afraid of a trial being negative. Like, everyone wants the trial to be positive and they want the technique to be good. And that's the problem we have as surgeons, because we have an inherent bias. You start the trial with what you want, what you think should be the outcome, basically. And you're doing the trial to prove what you think is right. This is not a way to do research, in my opinion. Research should ideally be neutral, and then you should come to present it. So the beauty, I think, of international collaboration is actually opening it out to getting that breadth of diversity in terms of your patients, but also in terms of your surgeons. And I think one of the things that we should be less afraid of is actually using the trials as a vehicle to improve the quality of surgery internationally. So part of the work we do with surgical quality assurance is it's an educational platform, because you're going to bring 20 to 30 centers across the globe together internationally to deliver a technique. And you can start to define a standard for what you think is good enough, basically. And then that can actually raise the quality of surgery, not just within your country, but also internationally, which I think is an exciting opportunity.
Dr. Joe, do you have any questions for our panelists?
Yeah, I think I'm representing the kind of the basic researchers here. So, of course, like previously, we have really good collaborations with breast surgeons and the brain surgeons, and to collect the samples and run those single cell analyses and to identify more mechanisms of the immunotherapies. So I just want like a brief comments from both like Professor Marker and Redis, like which ways you think it makes more sense for the basic researchers to collaborate with the surgeons? Like, what, in addition to like just obtain the clinical samples from your guys, is there any other exciting opportunities that we can push the therapies forwards? Thank you.
Yeah, so I think for maybe start, but I think for us is that we have a lot of lab researchers in Oxford, obviously, and they come to the operating theatre. Like I insist, like I say, if you're going to do research in this area, you have to come to the operating theatre. I'll give you a chair if you're going to faint, but you have to stand there and watch an operation. You have to sit there or stand there and watch an operation with me. And I think it's very important for that aspect. I think also as well it should be bi-directional. So in so much as whilst I know, for example, in my own area that there's only a third of patients who will benefit from immunotherapy, but if we can actually create an iterative loop whereby we're able to, in a platform, ultimately make decisions very quickly, then it will provide an opportunity both from a clinical trial point of view, but also from a translational research point of view, to accelerate your translational research into clinical implementation. Rather than waiting for your 500-man study, you can power it on less than that, around 50 patients, and make decisions quicker and move along.
Professor Chu has excused himself because he has another conflicting meeting at the same time, so he will not join us in discussion, as I understood. But Mr. Reinders, any comment on Dr. Zhou's question?
Well, maybe I wanted to begin on what Professor Marker said. For the researchers to be acquainted with the clinical practice, it's not just for the researchers. It's also, for instance, for us statisticians, very informative to really know how things are done, how things run, how information is collected, because it all determines how we should interpret outcomes, and it helps to identify potential pitfalls that each discipline or each person and each discipline on his own may not have the capacity to detect, but if insights come together, we often come at better conclusions. So really like that remark.
Very important observation. Doctors are not so good with statistics, unfortunately. That's why we have teams in hospitals that help out. But we need a basic knowledge at least. Question for everyone. What do you think, which area of surgical oncology is currently most in need of high quality prospective evidence, in your opinion?
So I think it's hard to sort of pick a particular specialty. I think that's probably the natural thing. But if we're starting to think about, I mean, the big growth areas, I think, will be in immunotherapy, without a doubt, which patients, and it'll be on both sides of the scale in many ways. Those patients who have such a profound response to immunotherapy that they maybe don't need surgery anymore, and those patients who have a disease which would be classed as unresectable or metastatic disease, which then become resectable. So these are the two main areas. I think that the pendulum of where we operate will be very different in 5 years' time. The second aspect is in terms of pure surgical technique, is I think at the moment the evidence in favor of robotic surgery is very poor. I think there's a lot of work to be done. I really do. I think it's coming. Like I agree that it's definitely, the robot is definitely going to be is a major player. I do robotic surgery as well, but when I say to my patients in clinic, I say there's no great data to support this. Basically, in reality, we do it because it's basically easier, and I get to sit down. Basically, so I think, and that's the honest truth. And I think, and people who are searching for this tangible benefit with robotic surgery will struggle. I personally believe, but yeah.
I fully agree with you on this. It's difficult for people to become part of randomized clinical trials. It's even more difficult to run important trials. So how can an individual surgical oncologist become part of the evidence generation process? What can one person do? How can we contribute?
So I think just quickly, but I think it's just important for everyone to understand that you, by definition, you have your skills. So as Professor Renda said, statisticians have their skills. As surgical oncologists, we know the clinical area, you know the clinical questions. If you have ideas, these are because you know the area, you know the problem. So then the question will always be then is how far do you want to take it and how much effort are you willing to put in to try to get there? And that's where I think societies like ESSO are so important by trying to create networks whereby we can facilitate this. One of the things that you do is you reach out. So if there are any junior researchers on the call, I used to, whenever I went to a conference, I used to tag a day before or after and visit a surgeon and watch them operate, because the surgeons are most relaxed when they're operating. You talk to them about research, you talk to them about your ideas, and then that way you start to get involved and you start to understand what's going on in the field. And I think you just have to start somewhere. But the most important thing is to know your currency, which is you know the clinical problem.
Yes, and also international collaboration can be really important in that matter. Search for some mentorship in societies, for example, like ESSO, which provides mentorship and guidance for early career surgical oncologists and young researchers, and also provide fellowships, observerships that can all be very, very encouraging for young people to get involved. I think this is the best way to engage young surgeons in research and clinical trials, don't you think so?
Yeah, very much so. I think and I think just having a dialogue. I think starting a dialogue with people is important, really important, and lean on expertise. Like as you said, I think we have a very multidisciplinary team with several statisticians in my department, and I will go commonly and knock on the office and just ask them, yeah, okay, look, I've got this idea for something. What do you think? What would be the best method? And that started from when I was very young. Like I did exactly that with my friends who were doing mathematics at university. So
yeah. Connections are important, and some platforms for discussion of your ideas, and also medical statisticians that are supporting your hospitals. That's crucial, I think, especially for people who have very little experience. In ESSO, we have Research Academy, and we are trying to restructure it in order to be also very involving towards young people. To provide mentorship for people who have ideas, some good pitches, but don't know how to start with. And I think that's something that's important step for somebody in the early career. Asking our colleagues from China, what opportunities do you think we have for stronger collaboration between European and Chinese surgical oncology communities? Dr. Zhao?
So I think for example, my PhD supervisor, who's a brain surgeon at Edinburgh Hospital in University of Cambridge. So he always is surprised when he first visit Chinese hospitals. He mentioned to me there's just 50 cases every month maximum for brain cancers in Edinburgh Hospital. But for example, in Professor Pengfei's hospital, there's thousands of patients are diagnosed with brain cancers each month. So I think each multi-center studies has its own stress. For example, in China, it has the most standard medical therapies in the renowned cities. So I think with the high level of population which treated with the most updated medicines could be qualified for the trial to include this patient. So I always think the international collaborations is quite crucial. For example, there is an ongoing clinical trial between Cambridge and Shandong, which is focused on both the brain cancers and the breast cancers. So I think this is good, and this is maybe can open some opportunities to boost the trial to recruit more and more patients on a relatively shorter time period. So yeah, I'll just shortly comment on this.
Thank you. You need to step in for the Chinese community listening at this moment because Professor Chu is not available to comment. But thank you for your insight.
Actually, Professor Chu is doing another surgeries.
I
another...
told them that it's not possible to involve him in the discussion. Otherwise, we would obtain important insights from his experience. Let me ask one question to those two panelists remaining, and also anyone willing to comment. Looking ahead, what will be the most important change in the way we conduct surgical research? What is your opinion, Professor Marker?
I think we probably need to become more agile with surgical research. We're less patient as a international community to wait for results, I think. So I think we have a couple of aspects. One of this will be embedding trials within registries, and therefore it becomes more representative of the international population. So therefore the trial will have more external validity. The second aspect is that we'll probably start to use more surrogate markers for survival in surgical oncology. So whether this be resolution of ctDNA or regression of primary tumor, I think these kind of things will become more commonly used as surrogates for overall survival, whilst we await the longer term results, almost certainly. Once we've got valid, very well validated biomarkers for response, then I think this will probably become more common.
Yes, wonderful insight on this question, Mr. Reinders.
Well, I think over the whole landscape, not just the surgical landscape, the more complex trials, the platform trials, adaptive trials are getting more and more common, coming up with more diverse settings, more comparisons to be made, and often also quicker answers, or at least quicker early answers. So I think the more complex trials that can get quicker answers will lead to quicker answers and better research, as they can respond quicker to what is observed.
Absolutely. I will invite everyone from our Chinese participants today also to check out the ESSO website and maybe have some more insights in what we are doing, and also join and suggest some research because we have Research Academy and also availability for different collaborations. So you're free to check out the website and send your pitches and inquire about potential collaboration with us. Dr. Joe, do you have any comments before I close the webinar?
Actually, I have one last question regarding the usage of the multi-omics in for example, the biomarker research for the clinical trials. So currently, we are conducting several researches to utilize both artificial intelligence combined with single cell and spatial omics to predict the response to for different category of patients in one clinical trials. So how useful is your opinion, like in combining the AI and multi-omics, for example, the single cell and spatial omics in the real clinical trial biomarker research?
I think it's hugely important. I think it's hugely a massive area for growth, definitely for scientific growth, without a doubt. Question will always be with any AI algorithm is disparity, basically, and whether or not your AI algorithm's been developed on a population that is representative of the general population. So definitely it's something we should be doing. Definitely it's something that will come, but the population needs to be diverse enough to ensure that the algorithm has external validity outside of the population within the trial.
And then maybe add that the results of the biomarkers and how to act on it eventually will have to be tested in a more conventional way, of course, which will always consume resources and time. And so, yeah, the end stage will always be a slower stage there.
Yeah, thank you very much for your comments. Yeah.
Thank you. So we have reached the end of today's webinar. I think it was interesting and engaging. I'm sure that the audience was enjoying your talks. I'm not so happy that nobody asked any questions, but we had a few, so it's enough, and we try to engage everyone in more discussion that goes beyond your lovely talks. Just to put some closing remarks on today's wonderful educational activity, I think surgical innovation is global, but evidence generation must be standardized. Today's discussion highlighted that excellence in surgical oncology depends not only on technical expertise, but also on the quality of the evidence that supports our decisions every day. From preclinical discovery and translational research to innovative trial design and advanced analytical methods, every stage of the ideal framework contributes to improving patient care. As surgical oncologists, I think we have a large responsibility to actively participate in research, to foster international collaboration, and to generate evidence that is both scientifically rigorous and scientifically and clinically meaningful. Only through such efforts we can continue to advance our field and deliver better outcomes for our patient, which is our end goal always. And one special final note to young surgical community present online, it's not important whether you are leading multi-center randomized clinical trials or not, because almost everyone can contribute through prospective databases, collaborative studies, translational projects, methodological rigor, and participation in international networks such as European Society of Surgical Oncology. So I invite you to join us and to check your options and potential for the career growth within ESSO. Thanks once again to all the speakers and for fantastic panel discussion. Thanks to our Chinese colleagues for putting up this interesting topic of the webinar, and I'm giving my regards from Serbia.
Thank you. Thank you very much, Nada, for your moderating, and thank you for all the speakers. And I think we will have more opportunities in the future to collaborate. And since we organize a group of Chinese scholars and will attend this year's annual meeting in Madrid, I think we will have a deep collaboration plan at that time, I think. Thank you.
Wonderful. We're expecting you in Madrid.
Thanks you.
Thank you.
Thank
Bye,
you so much.
everyone.